Welcome back, everybody. This is chapter 25. In this chapter, we're going to be talking about microbes in the environment, as well as the concept of One Health, talking about the overall health and well-being of all people that reside on the planet altogether. So let's jump right into it. These are your learning outcomes for this chapter, for those of you who are keeping up with that. These are concepts, questions that you should be able to answer. So microbial ecology is a term that refers to the study of microbes in their natural habitats. We are able to study them a little bit more detail because we do have genomic techniques that allow us to sequence the genome of these organisms. So that's been helpful for understanding a lot of these organisms. So the importance of microbes, what role do they play in the biogeochemical cycles? And you might have been, just losing sleep over wondering what they do with regards to the biogeochemical cycles. I know I do. So this is referring to basically how a lot of the elements in our environment are going to be cycled within the microbial population, and therefore what we would depend on them for doing, recycling those elements in ways that we would not be able to recycle otherwise. So the elements that we would be concerned about how they are cycling through the environment would include carbon, nitrogen, sulfur, phosphorus, oxygen, and iron. Those are some biggies there. Ultimately, they're all originating. That's a weird way I said that. Originating from a non-living source. So, whether it's from rocks or gases in the atmosphere or any of that. So we can cycle these things in their pure form, elemental form, like N2, that's just nitrogen, right? Or as a compound. So that would be phosphate. So phosphate is phosphorus compounded with, of course, oxygen atoms. And we can cycle these things through redox reactions, so exchanging of electrons. So elements can cycle between non-living and living environment. We know this because we can eat animals or plants or other organisms as a food source. And then even our waste can get reabsorbed into the environment thanks to being broken down by microbes. So a lot of that is really important as far as recycling nutrients in the whole environment. These cycles often are, going to be circular in a way. So the waste products of one organism, like I was just saying, could be an energy source or building material for another organism, like our waste would be for bacteria. All of the organisms in the entire, Earth biosphere, all organisms will participate in some way in recycling these elements, but just depends on how, from organism to organism, based on whatever metabolic pathways they can follow and what form each of these molecules will be taking as they're getting cycled through those organisms, of course. So in the universe, we can look at geomicrobiology. Here we're talking about microbes in a geological process. So whether that is maintaining the Earth's crust, being involved in formation of fossil fuels. And then we can even look up into the stars and the study of what we believe life on other planets would look like. That is astrobiology or exobiology, same thing either way. So there are people who do study that and do major in that in college. So looking at the carbon cycle, we know carbons are fundamental molecule for all of the biomolecules. We're talking about organic molecules in general. So it's one of the largest in our biomass. This will exist predominantly in a mineral state, and there is an organic reservoir in the bodies of organisms and like carbon dioxide in the atmosphere, methane in the atmosphere, and those sorts of things. Those are inorganic forms, right? So yes, so let's go through it. We know that this is a plant. So it is obvious. Oh my gosh, I forgot again. Sorry. We know that plants perform photosynthesis, and that in the process of performing photosynthesis, they will be able to fix carbon dioxide from the atmosphere into glucose, which is an organic molecule that can then be stored or used by the plant as a means for creating energy through aerobic respiration, just like we do. And we can also eat these plants and then gain the carbon from the organic molecules that were used by the plant to build its plant structure that it got from, breathing in, quote unquote, carbon dioxide from the atmosphere. So that's pretty cool stuff. So that what we have are free carbon dioxide in the atmosphere, and now it'll be used up by the plant and fixed into organic molecules. Whether it's carbonate in the sediments out here, so that is also carbonate being an inorganic source of carbon. Whatever it is, it could be from the plant itself, from it dying, or it could be from inorganic sources from the sediment. There are microbes that are able to utilize these as sources of carbon. So whether, like I said, organic from the plant or inorganic from the soils. And then those can be eaten or whatever, broken down by other organisms that are larger in the food chain, and so on and so forth, and including us. Of course, we would fall here as some animals that would be involved in utilizing the fixed carbon organic molecules that we were seeing in the plant. And the saprobes would then break it down after it died. That's what these are, organic carbon being broken down, decomposed. Those are saprobes, right? So anyways, their aerobic respiration will release carbon dioxide in the atmosphere. We already know this because when we convert pyruvic acid into acetyl-CoA, we release carbon dioxide. We also release carbon dioxide during cycling through the Krebs cycle. So anything that's going through aerobic respiration is going to release carbon dioxide back into the atmosphere, and we can start the cycle again. We also have combustion, whether it's natural or like we would see in a volcano, or created by man, either way, releasing carbon dioxide into the atmosphere. And over time, we have seen a massive, massive increase in carbon dioxide in the atmosphere. This is no longer, something that anybody is questioning. I'm just saying, if you are, then you are way late to the party. The introduction of so much carbon dioxide into the atmosphere has made a massive, massive impact on our environment. This is what we call, climate change or global warming, but these are real events, no longer refutable. I'm sorry, but they just aren't. Then we have methane. This is also a very important greenhouse gas. It's actually going to trap more heat in the atmosphere than even carbon dioxide would. Methanogens are methane producers, like in our anaerobic swamps and things like that. Those guys are going to be the primary producers for that. We do have animals that produce it as well. So that's going to be about 20% of the methane in the population, thanks to those guys and agriculture. So we have in the nitrogen cycle, we have 4 basic kinds of reactions that are going to be going on. It's basically cycling nitrogen in different forms through plants, through the soil, and back into other organisms. And that is nitrogen fixation, ammonification, nitrification, and denitrification. So try to see if I can keep up with this. So, okay, so denitrification. So I can keep up with the order of these as we're moving through, even though I think it's going to say, I don't know that it does. Okay. Root nodules, very, very important as far as supplying nitrogen to the plant. Why does the plant need nitrogen, by the way? That would be our nitrogenous bases in our nucleic acids, as well as our amino acids, that amino group, the NH2 group, that involves nitrogen. So we have the involvement of rhizobia, which are gram-negative bacteria, getting involved with the root system of these plants. And those bacteria are going to create a symbiotic relationship there that provides nitrogen to the plant so that it can live and grow. So and ammonification, here we have decomposition of organic matter. What did I say? I said decomposition. I hope that's a word I used. I don't think it was, but decomposition of organic matter by Clostridium and Proteus species. So that's ammonification because they're producing ammonia as a byproduct, essentially. So, so that's going to be the decomposing one. Okay, then we have nitrification. We have organisms that are going to oxidize NH3, which is that ammonia-based molecule, to NO2, whether we're talking about nitrite in that case or nitrate in the NO3 situation. So that's going to be nitrification. So we have our NH3 to NO2, nitrite, to NO3, nitrate, back to nitrate. Okay, so this is a breakdown of those. For denitrification, we have the conversion of that NH3, that's that sort of ion created from the ammonia. So we're creating an ion, and we can go through steps. Okay, sorry about that. Denitrification is basically going to be dealing with the ammonia ions and turning them through several steps back into N2 gas. So N2 is just atmospheric nitrogen. That's essentially what that's going to be talking about. Okay. Okay, so we have microbes on the land and in the water. We're looking at the Earth Microbiome Project to understand where microbes are found in the atmosphere, in the biosphere, the livable areas of the Earth, essentially. So there's all over the place, right? We already know that. That's no surprise there. So metagenomics is just taking an environmental sample and then just doing shotgun sequencing and seeing what you get. That's basically how you can identify a lot of different organisms in a lot of different environments, and just taking pieces and pieces and pieces and going through PCR based on that sequence, and then, whatever, getting our results and being able to compare it to organisms that we already have sequenced, or organisms that maybe it's a new organism. So as closely as we can get to an existing species. Then comparing from there, we can determine what their genes might be able to do. In soil microbiology, we already talked a little bit about this, the importance of bacteria as far as providing that the nutrients to the plant roots. So that's important, of course, as far as nitrogen goes. But then we also have other types of organisms living in the soil, aerobic and facultative organisms in looser soils, or anaerobes in the waterlogged, poorly aerated soils that are going to be producing and interacting with all these other organisms there. So the rhizosphere is the zone of soil surrounding roots and plants. It contains bacteria, fungi, and protozoa. And there's probably viruses involved in that as well, FYI. And then we have and how they interact with the plants that they are growing around. Often this is a symbiotic relationship between many of these organisms. So it creates this sort of micro community. Mycorrhizae, this is a mutualistic partnership between plant roots and the microbes. Essentially, the plant feeds the fungus through photosynthesis, and then the fungus helps anchor the plant, as well as capturing water and minerals for the plant and supplying it to the plant. We have sampling of deep subsurface microbes 2 miles or more below the surface. There have been bacteria found 30 meters below the deep seafloor in clay deposited 86 million years ago that were undisturbed clay. And there's still microbes there that are still alive. Undisturbed, I want to make that clear. Undisturbed, living in there for 86 million years. And yeah, so it's pretty interesting stuff, and it can give a hint as to the origins of life. As far as the importance of water, we all know that water is the solvent of life. So when we get into talking about water, we need to consider, maybe where our water is coming from in the large scheme of things. So we can break this down as most of that water is going to be saline. We already know this in the oceans. Yes, that freshwater sliver that's there, break it down further into things that are going to be existing in ice caps and stuff like that we wouldn't necessarily be able to use. And then if we break down the stuff that we can use from the fresh surface water, then that's going to include our lakes and our rivers and our swamps. And that would be what we could, potentially consume. But yeah, so that's where our water is. A very, very, very small percentage of it that we could actually use. So anyways, the surface water is evaporated. We know that enters the atmosphere and falls as rain typically. And then that will create runoff, and then, whatever. And through all of that, we will have introduction of that water that has been introduced into that system. and is running through that system, introduce it to aerobes that will give off water through respiration as a byproduct, which we already know, and run off as into the oceans and stuff again to start the whole cycle, right? An aquifer is a deep underground source of water that circulates very slowly. It is an important source for replenishing surface water and resurfaces through things like springs. It's the primary supply for about 1/4 of all of the water that people use. Okay, so very important. Then we have this guy that sampled a bunch of genes in an ocean and found out all these new proteins and all these new genes that we had never seen before. And it just puts into perspective the biodiversity that is out there that we just aren't even aware of. In marine environments, we can also have things that are ultra salty. So we have freshwater, we have marine, which includes just saltwater environment. And then we have the high salinity things like the Dead Sea that you can float super easily on. People go visit and want to have like the minerals from the mud or whatever is apparently good for your skin. I don't know. Then the salinity of that is going to be at 35%. So we consider that to be hypersaline. So very, very salty. Scientists have found that there are microbes living in here. They are salt tolerant archaea. So we know about those extremophiles. There are bacteriophages in the ocean. It's kind of like what that guy was talking about with all those different genes and everything. We found 10 million viruses per milliliter. A milliliter is a cc, just to be clear. 1 milliliter, 10 million viruses floating around. Most of these are going to be bacteriophages. Yeah, and so there's a lot more out there than what we even know about, which I keep saying, but it's true. So some environmental factors can cause an increase in the number of algae that can cause, so these algae are normally food for other things in the food chain. However, certain changes in the climate can lead to overgrowth of these algae, and they can produce toxins that can then harm fish, shellfish, and even humans, and they can't be controlled. And that can lead to development of a potent mussel toxin that can be concentrated within the shellfish that we can then consume and then cause problems for us that way. So those are algal blooms or red tides. So those can be very dangerous for all sorts of animals. The plastosphere, this is a pretty serious problem. Most of us are aware of this to some extent, but maybe you didn't know that 88% of the ocean's surface is coated with microplastic particles. So when I learned about that, when I read that, I was actually pretty shocked. I didn't realize it would be 88%. Yeah, so we're mostly talking about microplastic, right? And a lot of those are the little beads that used to be included in like, hand wash and toothpastes and all sorts of things like that. So very small pieces, but still they are not supposed to be there. That's not part of the ocean. What's the problem with all of this? It does create a reef of sorts for certain microorganisms where they shouldn't be concentrating. They shouldn't be able to concentrate in these areas. It also obstructs the sunlight from reaching the ocean and creates a health risk for invertebrates and fish in the area. All right, let's talk a little bit about more of so this is talking about plankton and it's talking about freshwater communities, but we do know that plankton exists in the ocean. I hope you guys are aware of that anyways, but the plankton that are living in freshwater communities, these guys are going to include our phytoplankton and things like our photosynthetic algae and our cyanobacteria. These are photosynthetic, creating their own energy from the sunlight, and they're a huge contributor to the oxygen in our atmosphere. And then we have zooplankton, which are microscopic consumers. So they are not producers. Producers produce the energy. Consumers consume the producers. So that's the idea of how plankton essentially supports all of the organisms in those types of environment. There's not very much oxygen in water. It's typically used up by plankton, especially whenever they're going through aerobic respiration. So concentration will form a gradient, highest in the upper area and lowest in the bottom, although in the warmer strata on the surface, there can be lower levels of oxygen. That's typically where the organisms are living and using up the oxygen. So that can depend on temperature there as well. So oligotrophic versus eutrophic. Oligotrophic is talking about nutrient deficient aquatic ecosystems. The hyphomicrobium and the caulobacter organisms. I don't know much about these guys, obviously, but apparently they capture amounts of hydrocarbons in these environments and are able to use them for their own sources of energy and nutrients. But so that's the oligotrophic areas. The eutrophic, so this is going to be eutrophication is addition of excess nutrients to aquatic ecosystems. So this is usually going to be involved with triggered by, as it says in the bottom, usually associated with increased waste introduction into these environments, usually like we're talking mostly about lakes here, detergents in wastewater, runoff of manure, and all that sort of stuff. So that is going to be eutrophic. So too much nutrients of maybe not even the right kind, what it would favor ultimately, and instead is favoring the wrong sort of organism. So aerobic heterotrophs will deplete the oxygen further in that upper layer, right? So that'll decompose by decomposing organic matter, and that'll lead to die-offs because they can't, like the fish need oxygen to live, right? So we're using up all that oxygen. Those heterotrophs are going to overgrow due to, let's say, the eutrophication and cause the death of those fish and other things associated with that. Anaerobic and facultative microbes will survive this, but this can be remediated, and it will be by the biosystem itself over time, given time, right? So it can survive, and these things are things that happen naturally as well. It's just a lot of them, a great majority of them lately have been associated with human introduction of these things. So biotechnology, we've really mostly talked about the importance of microbes and what they can do for us in biotechnology, but really looking into all this, we can talk about decomposing things for industry. We can talk about fermentation of wine. We can talk about beer. We can talk about bread dough, DNA technology, food sciences, and all sorts of productions of vitamins and things like that by these organisms for us. And we can utilize them or hijack really their systems to favor our goal things. So fermentation, we can use this for mass controlled culturing of microbes to produce our organic compounds, whether that's acid or whatever it is that we're looking at, that we can grow them en masse to create whatever that is. So whatever you're interested in, whether it is acid or whatever. So obtaining this, we would use this to obtain many food and industrial products. That whole concept behind harnessing what is happening with fermentation. This includes making beer. This includes making bread. This includes making wine. All of these are going to be utilizing that concept of fermentation and growing microbes in culture in mass in order to get whatever product we want. So that's useful for us there. Moving on to drinking water. Like we had said, we were talking about aquifers and rivers and springs here. You're not really going to find it in its purest form nowadays. Most of the time it's going to have been filtered or something at some point. So treated to make it friendly for us. Right. So typically how this is going to work is we'll have that raw initial water that we would be facing. We'll put it through coagulation to try to make any particles that are in there to stick to one another so that we can cause the sedimentation of those things, so that they will fall out of solution, and then we can take, the top portion of that and then filter it to remove any other particulate matter that could be in there, and then we can disinfect by introducing it to UV light and treating it with chlorine gas, and then distribute it to people after that. So that's the idea of how that would work. In many parts of the world, sources of drinking water are also used as a dump or solid or liquid waste. What we're saying here is we're dumping sewage in a lot of certain areas of the world in the same places where children are playing, where people are washing their clothes, where they're getting their drinking water from. So used wastewater draining out of homes and industries can be a problem. We have in that sewage, we have chemicals, debris, and microorganisms, of course, and then large amounts of solid wastes, dissolved organic matter, and toxic chemicals. It's not just going to be poopy water. There's other dangerous things that are associated with this as well. So to separate out, to treat our wastewater before we put it back into rivers and stuff where people are actually, splashing around. So that's what we do with it, by the way. We treat it and then we put it in, but we test it first. We do in the, first world countries. So here's the process involved in that. The primary stage separates out a lot of the matter, the larger stuff, as well as skimming off the smaller stuff and allowing everything to settle. And then we can go through the secondary stage where we will biodegrade any organic matter, thanks to bacteria, algae, and protozoa that will break all of that down. And then sludge will settle out, and that will be involved with aerobic decomposition of that sludge, breaking down as much of it as we can. So things like wood, paper, fabrics, petroleum, and organic molecules. And then the tertiary stage, we have actual filtration and then chlorination. So we've broken down all of the matter, especially the organic stuff. We've removed the larger particulate that we wouldn't be able to digest otherwise. And now we are moving on to the filtration, chlorination, and then discharge. We can use this water for watering golf courses and watering parks, for releasing into larger bodies of water, like where they came from. And the solid waste can be used as well as fertilizer, and it can be applied to the land to help just the land recover from overuse or something like that. So that's essentially how wastewater treatment is going to work. So with COVID, this basically revived the practice of wastewater surveillance for pathogenic microbes, because we had to know how prevalent the COVID virus was going to be in our wastewater, and were we reintroducing COVID back into the system whenever we treated our water. And that was a big concern in China, I know. So we used this whole concept with polio, a much, much smaller virus, by the way, which is waterborne. And that we use the concept of how to clear out polio from the system to help clear out COVID from the system, essentially. So the virus is shed in fecal matter a few days before symptoms appear. So you might not even know that you were secreting it that way. And its presence can be monitored in the system during the wastewater treatment. But this will not distinguish between symptomatic and asymptomatic. We're not going to, it doesn't matter. You're still shedding the virus, essentially. But it is low cost, surveying this way, and will allow us to know which areas are going to be more likely to be positive for COVID, right? So we're actually looking for the RNA of the virus. So in the United States, hundreds of people develop waterborne illnesses every year. This is going to include things like cholera. It used to include something like polio, but we have almost eradicated that from the planet. Not completely yet, but we're pretty close. Right. So this is talking about depending on a clean and potable water supply. What does potable mean? Potable means free of pathogens, and then also of dissolved toxins, disagreeable turbidity, odor, color, and taste. Those are ideal situations, but potable means free of pathogens. And it might be cloudy, but as long as it's free of pathogens, you could technically consume it and not die of dehydration. So that's the idea there. So these are the waterborne pathogens that we might be concerned with: Giardia, Cryptosporidium, Campylobacter, Salmonella, Shigella, Vibrio, Mycobacterium, Hepatitis A virus, and then Norwalk virus. You probably heard of at least a few of these, right? So these guys, we want to be sure that these are not in our drinking water at any time. We don't want that. So what we do is we don't necessarily always test for these organisms, and particularly because we don't want to miss them in case there's just a few in there. So what we do is we look for indicator bacteria, and these are species that are intestinal residents of birds and mammals, and are going to be used for identification that if these guys live, then the other bad guys that we were talking about in the list prior to this, those pathogens, they could survive too. So this includes coliform bacteria. Those are just anything fecal coliform bacteria. That's what these are. So if they are from feces, then these other guys also live in the gut and could probably survive similar conditions. So if they made it through, we're going to assume that those other guys could make it through too. This includes E. coli, Citrobacter species, Klebsiella species, and Enterobacter species. Most of these are gram-negative. They are lactose fermenting, gas-producing bacteria. They are enteric, so from the colon. We use this to monitor essentially how pure the water is. Number of coliform bacteria commonly found in growing in fecal-free environments is something that we would want to monitor and keep an eye on. If it's not supposed to be there, why are we finding coliform there? That would probably be because our filtering mechanisms are not working properly. So microbiologists are now advocating that E. coli alone, just by itself, be an indicator for fecal contamination. Just because we don't want to rely too heavily on it having to have all of these things or anything like that. So that's what it is. I don't know. That's more or less, but bureaucratic stuff. I don't know. It's not going to concern you here. So water quality assays, there's one rapid method to do this. We're going to plate it on solid media, our water sample, so straight from your tap, for example. Count the number of colonies and estimate the total number of colonies that's present on that plate versus how many would be in your actual sample, your water sample. And this will not differentiate from coliforms or other microbial species. But if you have no colonies, then you probably don't have coliforms, right? That's the idea there. So it should be nice, clean, no colony, ideal situation. It's not what it is in reality, but that's because of pipes and how things work. In pipes, there's stuff in there. Sorry to remind you of that, but there is. The level of dissolved oxygen, we can also look at that. Water that contains high level of organic matter and bacteria will have lower oxygen due to the metabolism of those aerobic organisms. So if it had lower oxygen levels than what we were expecting, we would assume that it is contaminated with something. There's a membrane filter method where we're going to filter the water through this actual literal filter, and then basically put that filter onto a Petri dish and grow to see if there's any colonies that pop up. So this, again, can allow us to count non-fecal and fecal coliforms so that we can identify, how many colonies we would get growing on there, and then determine, okay, if we filtered this and these ones got stuck on the filter and they grew up and had these colonies, then this was 1 milliliter or whatever or 50 milliliters that we filtered out. And so this is, 50 colonies per 50 milliliters. And then what would that mean for the overall water supply? So that's the idea of how that works. So coliforms, we'll refer to the number as the most probable number. So this will detect coliforms. We're going to count the number of them and using whatever system we just talked about previously, and determine that number as presumptive, confirmatory, or completed. So you would want to go through several levels of testing in order to confirm whatever values you're attributing to that water sample as having that number of coliforms, or whatever sample you're looking at as having that number of coliforms. And then we can evaluate tubes for gas production, since most of these guys are gas producers. The number of positive tubes will be tallied, and then that will be applied to a statistical table, allowing us to determine the probability of coliforms in that sample. Okay, so microorganisms are also useful in our food. They're not just poopy. So we also have microorganisms that are involved in helping create kombucha. If you guys have ever drank kombucha, there's usually a mother involved in that. It's this like thick layer of bacteria and stuff, essentially, that's going to cause, it's sort of like the mother in the apple cider vinegar, if you know what I'm talking about, but it's the stuff that was involved in the fermentation process to give the fizziness and the sort of acidic taste to the kombucha while it was fermenting, because it's fermented. Same thing with pickled cabbage, Norwegian fermented fish, and Limburger cheese. These are pretty rough things, right? Pretty strong flavors. When we say pickled cabbage, we're talking about sauerkraut, just to be clear there. So these can give quite a unique and delicious flavor to some things if you are into some of those flavors. All right, so food fermentations, like we were just talking about, we will actively encourage biochemical activities that impart a particular taste, smell, or appearance to those foods. If we are going for a goal, will help treat the environment that those things are growing in order to favor those tastes. The microbes can occur naturally on the food, so they can, and the ones that are, what I'm saying is microbes that occur naturally on the food that might not be the fermentative ones necessarily that you're interested in. You might introduce special fermentation bacteria, but the ones that are already normally associated with the food will be included with the process, and that helps aid to different flavors as well. And so that is what causes things like starter cultures to differ from one another, and why they're not the same always between certain things, as well as the environment having quite a lot to do with it. Bread that is made in San Francisco, sourdough bread in San Francisco, is completely different flavor than sourdough bread anywhere else pretty much in the world. You can't get that same flavor anywhere else. You can't. I'm sorry. If you think you can, you are wrong about that. Unless you're buying it partially made or something like that, then if you are, buying it and it wasn't made in San Francisco, then it doesn't taste the same. I can attest to this, having family and been to San Francisco for many, many years. I'm just saying. I'm a little bit of a snob when it comes to the sourdough. All right, so many food fermentations are synergistic. That just means that, a lot of the bacteria that are involved in it are working together to create the flavor. That was a long little drink. Sorry about that. Okay, so bread making. We're going to be using microbes for leavening the flour-based dough. A lot of times we're talking about yeast here. The yeast involved in this is Saccharomyces cerevisiae. So you might want to be aware of that one. All of the leavening aspect of what is going on with the bread, all those air pockets and everything like that, is achieved by gas production during the fermentation process by this yeast. It provides flavor. It provides what they call volatile organic acids that give a certain smell to it, and as well as the alcohols that are produced as a result of it. So that's what makes bread taste like bread, honestly. So that was thanks to that yeast. So we can also condition the dough to make it workable. There are microbial enzymes that can help break down the proteins to give the dough a little bit more elasticity. It just depends on what you are introducing into your dough. So with beer, there's all these steps in beer, and I don't know much about it. I'll be completely honest, but I'm going to read it to you off of these slides anyways. All right, we have anaerobic fermentation of carbohydrates in fruits and grains that we will produce ethyl alcohol. That part, we understand that, right? Whenever we ferment something, one of the byproducts you can make is ethyl alcohol, ethanol, that's drinking alcohol. And then our byproduct also can be from that carbon dioxide, which gives a fizziness to that beverage. And that's where the fizziness of beer comes from. So yeast plus sugar. So that's going to be aerobic respiration is what we're talking about here, typically. But now we're going to put it into a fermentation scenario. So we're not providing oxygen, no more oxygen. So yeast plus sugar in this case, now we will go through glycolysis. We will cut off, we will not go into the Krebs cycle. That pyruvic acid that we made from our glycolysis, that will have to be converted to other organic molecules that will serve as the final electron acceptor to our NADH and turn it back into NAD to be reused in glycolysis again and again. So that's the idea of how this is going to work. We'll make carbon dioxide as a byproduct of all of this processing. And in this case, with these microorganisms, we will be making ethanol as a product as well. So that's fermentation. That we're going to force that fermentation to happen due to a certain amount of a certain lack of oxygen. So you can't have too much oxygen being introduced into the system. Otherwise, you're going to get aerobic fermentation and you will not produce any ethanol. It's going to fail. It's going to fail you. So our alcoholic beverages will vary in alcohol content and different flavors depending on whatever materials you're using and whatever organisms you're using. So let's start with malting. Here we have sprouted barley that will be softened, usually with moisture, to serve as nutrients for yeast. then that will release amylases, which are just enzymes that are going to convert starch, and to dextrins, which are just breaking product of breaking down starch, and maltose and proteases that will digest certain proteins that are available in the solution. So whatever it is, the fruit or whatever it is, breaking down all these different parts of the cells, right? So other sugar and starch supplements can be added, and then after the sprouts have been separated, the remaining malt grain will be dried and stored in preparation for actually mashing that. So that is malting. So we've taken the barley, we've treated it, we've digested it essentially with some of our microbes, and gotten it prepared for the yeast, which we're going to get to in a little bit. So so we're going to dry that and store it. Our malt. So then we have the mash. We have the malt grain from previously, soaking it in warm water. We're going to grind it up. Sugar and starch supplements can be added at this point as well to, favor whatever flavor or treatment you're trying to achieve here. We'll heat it to a certain temperature, which is here in this case 75 degrees Celsius, which will stop enzyme activity of the organisms, and then solid particles will be filtered out. Okay, so wort is the clear liquid that will be left after the mash is filtered. So we did the mash, we did the malting, we did the mashing, and then we have the wort. So now the wort is that liquid. We will boil it for 2.5 hours with hops to give it aroma and flavor. This will also caramelize the sugar, impart the color, and then get rid of any contaminants that could be there. And then fermentation, this actual fermentation step that's actually going to happen leading to actual fermentation. We will inoculate that wort that we've just treated that whole way with Saccharomyces cerevisiae and certain strains of that yeast that will be developed specifically for beer making. And then wort sugar, whatever sugar is in that wort is what it's saying, is converted to ethanol and carbon dioxide. And that will, stop once we get 3 to 6% ethanol. It won't go past that because the bacteria won't survive living in too much alcohol, right? That makes sense. You clean them with the alcohol too. So they'll stop after a certain point. All right, so that is that for the beer making. Next is the concept of One Health. This is just the health of all life on Earth is connected, and how the health of some organisms could affect the livelihood of other organisms, right? It captures interrelatedness of nature, microbes, the environment, and animals, including humans. So mixing of microbes in different animal hosts and under different environmental conditions can lead to evolution of new and potentially dangerous pathogens. Which that makes sense. We're already aware of how true that is in the case of the flu continuously, pretty much, right? So behavioral changes in animals and humans, environmental changes, and all of this stuff can lead to changes and cause the development of new infectious diseases in new reservoirs and all sorts of stuff like that can lead to stuff that we would no longer be able to predict as far as that disease is concerned. Changes can cause microbes to increase or decrease their virulence, so how much pathogenicity they have, how much disease they're causing in you. So even if you think you know these organisms. Hantavirus has been known to cause hemorrhagic fever with renal syndrome. It also causes hantavirus pulmonary syndrome. So that's more common than the hemorrhagic fever with renal syndrome. You don't want either of these, okay? This has a 50 to 70% mortality rate. That's no joke. It is transmitted by mouse urine and excrement. So this is essentially talking to us about, so we have, I don't know, pinon nut. Okay, I guess that's kind of nut. So this we had increased growth of the pinon nut. I guess it's some sort of food source for mice. And there was a lot of rainfall in the Southwest, which is normally dry. And so because of the rainfall, now we have all these nuts, and now there's more mice, and now they're going into the house, and now they're pooping and peeing everywhere. And then you go and you sweep it up to try to contain all this poop and pee that's everywhere suddenly. And now you're breathing in the particles from that. And now you get hantavirus. And now you have a hemorrhagic fever with renal syndrome, and you're going to die. So that's how hantavirus works. It's terrifying. It's one of the viruses I used to study when I was in graduate school. All right, HIV. yeah, we're all aware of this, but if you're sitting here being like, oh, boring, then you are not paying attention to HIV. HIV is a terrifying disease. Yes, we are managing it okay here in first world countries, but that's not where the concern really is. So most likely first humans were infected with the virus when butchering primates. This is similar to what we have seen to be the case with Ebola. Anyways, this is originally simian immunodeficiency virus and now has become a human immunodeficiency virus. So as it was transmitted to people, it became more virulent. So it has a worse effect on us than it does on the chimpanzees. So that's pretty, like I said, terrifying. I mean, how would you know about this? So sexual mixing in large cities allowed it to become established in Central Africa, and use of needles in large-scale vaccination campaigns also caused spread in it when people weren't paying attention to reusing needles because they may or may not know any better. When we consider the likelihood of being HIV positive in South Africa, it's something like 1 in 5 people in South Africa are HIV positive. Think about that. So there's Africa and there's Kinshasa, which is, I believe it's the capital of the Democratic Republic of the Congo. But anyways, that's where we think a lot of this originated to begin with. So we know that this entered the population in the 70s and 80s. The first cases were exclusively gay male community. They called it GRID, which was gay-related immunodeficiency. We now know that it's not because of being gay, but that there are sexual activities that gay males may engage in that could put them at higher risk for contracting the disease. And that's more or less what that's associated to now. Virulence of the virus has continued to increase over time. And obviously, heterosexual encounters can transmit the virus as well. There are other mosquito-borne diseases that are still concerning due to a change in the environment and climate and everything like that. Mosquito-borne viruses like Zika and West Nile virus, these are things that we had never seen before. And now as the climate has warmed, we've seen a massive increase in these things, hand in hand with the Aedes genus mosquitoes that are expanding their reach because we have these mosquitoes all over the United States and can be transmitted to people that way. Right. So there's an increase in the number of days mosquitoes are active now, and they're therefore able to transmit the disease. That's due to the climate change as well. And viruses can find new hosts. They live longer. The mosquitoes live longer, and so they are able to transmit this to more animals, and then more hosts are able to, maintain it. And these are just talking about the transmission of the disease over time. From what we saw in the 70s, based up to 2017 in this case. So then we have the emergence of COVID, of course, in 2019 in Wuhan, China. We believe it's associated with a bat virus that jumped species to another animal and then to humans. Bats are rich reservoirs of viruses, and they often live in colonies, so they can maintain the persistence of those viruses quite easily. Bats are known to transmit rabies, Ebola, and the first SARS to people. Bats are also regularly trucked to the wildlife markets of large cities, including Wuhan, which can lead to transmission, easy transmission from area to area. So that climate change helped support the transmission of this virus. We also know that climate change has led to quite an increase of wildfires in the West, especially in California, because of drying out in these areas. And so that effect that it is having on that environment will also change, essentially the organisms involved in those environments. Here is a little table summarizing some of the other effects of climate change, whether it be weather extremes, record high and low, allergen levels, rising sea levels, variable rainfall patterns, and then water and mosquito-borne diseases. I'm not going to go through this table right now, but you are more than welcome to take time to look through this on your own. I think it is important to be aware of these things in our environment. So these are some pathogens that are newly recognized in recent decades, just in the past few decades have been discovered. So some of these are very, very scary diseases, and you probably don't even know about how scary these are. Of course, we know about how scary Ebola is. I don't need me to tell you about that. But if you look up something like Nipah, which I recommend that you, that was a really good star, I recommend that you do, or MERS, which is very uncommon, but still I would look it up anyways. These are some interesting ones. Rift Valley fever. This is not as concerning for people, but it could destroy the cattle industry in the United States, and that would destroy the United States. So, but yeah, so some of these are newer. And then here are some re-emerging pathogens, things that we saw backing down and they weren't quite as common anymore. And now they seem to be peeking their heads back out as they're finding their niche once again. So, so we are seeing an uptick in Clostridium difficile, or C. diff, you know, pseudomembranous colitis, essentially. So that's due to having to reach for different antibiotics to control infection as antibiotic resistance is coming up. Staph, huge problem. Again, these are going to, and strep, both of these are going to be dealing with antibiotic resistance. So, so there's those. And then we have things like, well, okay, well, this one, Crimean Congo hemorrhagic fever virus. This is a disease that I focused my research on when I was in graduate school, the one I actually focused on. And this is transmitted by a tick. And they're hard ticks, they're exotid ticks is what they're called. But the change in the climate is causing an increase in, you know, the number of tick bites and transmissions, and people going out and getting bitten by ticks, and also the prevalence of the ticks themselves. So that would cause an uptick in this disease. And that's things that people don't think about. And of course, we know monkeypox. So these are some of the most concerning emerging pathogens for the World Health Organization, things that they are concerned about as far as why they're increasing. Like I said, Rift Valley fever, this is related very, very closely to the Crimean Congo hemorrhagic fever. Again, I studied Rift Valley fever. I studied all of those Bunya viruses. So includes hantavirus. But anyways, these guys, the Rift Valley fever, this is one of those diseases that is 100% abortive. And what does that mean? It's exactly what it sounds like. If you get this disease, whenever you're pregnant, you will abort the fetus, 100% chance. This is true for people, but it is also, this is mostly a concern for cattle. It's where this has been the biggest issue. So in Africa, where this is seen naturally, I even have a student that actually grew up in areas where they had this. This is a massive concern, especially with the increase of the mosquito population due to the warming climate. We have increased incidence of Rift Valley fever. And if we cannot keep this out of the United States, which we have been able to so far, but if it were to come into the United States, it would be absolutely devastating for the cattle industry here, because it is highly transmissible. And the mosquitoes that carry this virus, we have mosquitoes that can carry this virus here. So we don't want it here. All right, so that's the end of this chapter. So explain to me the importance of monitoring indicator bacteria like the fecal coliforms in water sources. And I will see you guys next time for the next chapter. Oh, I can click this, I promise.